This video is going to be about proteins and how their amino acids and the order of those amino acids dictates the shape of the protein, and we'll learn why that's so important to cells. Take a look at this picture with me. Here it says primary structure, and what we're taking a look at here is this chain of amino acids. Okay, so you can see here it says gly, pro, thre. So it's glycine, proline, threonine, glycine, thre. You guys get the idea. These are abbreviations for the 20 amino acids. And what you can see is that sometimes they repeat, just like letters of the alphabet, sometimes they repeat. And we know that they're in a particular order here. That order is dictated by the DNA. Usually when we draw an amino acid, you guys are used to seeing it like with a carbon in the middle. An R group, a hydrogen here. Then you have this carboxylic acid group here. And over here you have this amino, see it's written here, this amino end with these hydrogens. Okay, so this is just being represented as a ball with an abbreviation in it. Okay, and what primary structure is it just means that we're just showing you or what the primary structure of a protein is it's just the order of the amino acids. Okay, so it's like, well, first you'd have a glycine, and then a proline, and then a threonine, then a glycine, then a threonine, then a glycine. It's just the order of them. They're always going to have a linear shape, almost like if you've ever made a bracelet or a necklace out of beads. Okay, you would put like, a purple bead, then like a green bead, and then another green bead, and a yellow bead. It would just be like, oh, here's my string. Here's the one end of my necklace, and here's the other end of my necklace. So primary structure just means just the order of the amino acids. Okay, so I typed this out here for you. I'll put this all up here. So primary structure, you guys know primary, we use that word a lot in biology, that just means first. So it's the sequence of the amino acids that make up this chain. It's just what order do they go in. Okay, and then these things here, I think you already know this. This is a bit repetitive, maybe don't need to write that one. And I won't ask you this, but proteins are typically 300 to 500 amino acids in length. So that's what primary structure means, just the order of the amino acids. Now, we're going to see in the next level of structure, it gets a little more complicated than this. Okay, so proteins aren't shaped like a bunch of beads hooked together. Okay, they're not really shaped like that. Okay, but if we want to study, okay, well, what amino acids are there here? I want to know this one, then this one, then this one. Then we say, okay, now you're studying the primary structure. But in our cells, they don't just line up like a big long necklace. Okay, they typically fold a bit. And the first folding is called secondary structure. Okay, so you guys know secondary means it comes after primary. So what this is, you have your beaded necklace, and imagine if on your beaded necklace that like this was a magnet and that one was a magnet. And maybe that one was a magnet. And then the shaded in ones are actually little magnets you put on instead of beads. Well, what would happen is it would kind of crumble. Like those would attract to each other, and then it would attract that one, and it would kind of like fold up into like a certain shape because there's attractions between your beads. That's what secondary structure means. You have these amino acids in a certain line, in a certain order dictated by the DNA, and now they're going to start attracting each other and repelling each other because some of the amino acids are positively charged, and they'll attract to the nearby negatives. They'll also push away from the other positives. So positives will repel positives, negatives will repel negatives, positives and negatives, they'll hook together. And what happens is that it kind of bends a little bit. Okay, now these pictures might look a little crazy, but you guys know what a helix is. It's just a spiral. And what we see in proteins, if you look right here, sometimes we see a spiral in them. And the reason they spiral is because these amino acids attract one another in a characteristic way that we see a lot. Sometimes they make this kind of like bent sort of shape. Okay, that's what they're trying to show you here. It's almost like if you think of like a paper fan, how it goes like up and down and it has all these ridges. We see that a lot with proteins, is those attractions and repulsions make this, it's called a pleated sheet. Okay, it kind of makes these, think about like a paper fan is probably a really good analogy. So what this all means is secondary structure is when nearby amino acids are attracted to each other and they form hydrogen bonds. I'm trying to show you like right here, that's a hydrogen bond. And then right here, they're trying to show you that. Okay, right here we get some hydrogen bonding going on. So if you guys remember, hydrogen bonds are those weak negative and weak positives attracting one another. And it just bends that primary structure, that straight line, into these different shapes. And that's important for our cells. So I would think you would need all of these details here. So there's two more levels of organization. There's tertiary, which you all know means third level, and there's quaternary, which means fourth level. So let's get into tertiary structure. Tertiary structure, let me pop the definition up for you here first. This is due to bonds. So ionic, you guys know that's when electrons are given up and then received, and then it's a strong negative and a strong positive, making a very strong attraction. Covalent is when those atoms will share electrons to complete their valence shells. We already discussed hydrogen bonds, but guys, once you write this, this is the key difference. It's between distant atoms. So secondary structure was between nearby amino acids, and they would attract each other with hydrogen bonds. This is between amino acids that are far from each other, like maybe dozens of amino acids apart. So if you take a look, so let's picture there's an amino acid right here, and there's one right here. They're not really anywhere near each other. I mean, you get from this one, it's like, you know, following a maze on a piece of paper when you were like a little kid, right? Like, oh, look how far away it is. So yeah, this amino acid is nowhere close to this amino acid, but they're being attracted, and this thing they're calling a salt bridge is holding them together right there. Okay, that's helps make this tertiary structure. Same thing right here. This amino acid, if we pretend there's one right there, and this one are nowhere near each other, but they're being attracted and they're crumpling the amino acid chain. And that's what this tertiary structure is. Okay, it's almost like if you had a paper towel and you were to kind of crumble it up. That's what proteins really look like. So you see this word here, globular, and it just means they look like a blob. They look like a crumpled up piece of paper. If you look at, they make crystal images of what proteins look like. They all look like wadded up pieces of paper towels. Okay, and that just means globular shape. Now to our body, and to our cells, that wadded up shape, that globular shape, it looks like nonsense to us when we just look at these pictures. But to our body, that's super important. If that shape is off, like if this little bridge didn't happen the right way, this protein would have the wrong shape and it wouldn't work. Okay, not working means the cell's going to have some problems. So that's tertiary. So remember, primary is just the order of the amino acids. So and it never exists like that, but we could study it like that. We want to know the order. That's primary structure. Secondary structure is when nearbys attract and maybe like repel one another, and they make some of those alpha helices and those beta pleated sheets. Tertiary is when very far away amino acids attract and make some of these bonds and lead to this crumpled up globular shape. So just about every protein it exists normally as a tertiary structure protein, because that's how it has its unique three-dimensional shape that our cells need. And then finally, there's quaternary structure. This means fourth level of structure. If you take a look, this is a protein called hemoglobin. This is what's in our red blood cells, and this is what binds to oxygen. So when the blood goes through the blood vessels in the heart, in the lungs, it can grab oxygen, and then it can go down to like your pinky toe and drop that oxygen off. We'll learn about that this year. Look at this picture though. See how there's 4 different colors? This is 4 different tertiary proteins, but if you look, they're all hooked together and they make this massive protein. That's what quaternary structure is. It's numerous third levels, numerous tertiaries all linked together to make a bigger protein. Okay, and you could tell they have like different names. They'll call them like these different Greek letter chains and things like that. But that's what quaternary structure means. So let me pop this up for you. Okay, so it's a protein that consists of more than one. Polypeptide is a synonym for an immature protein. How a protein matures, if you guys think back to maybe a different class you had, um, once the ribosomes make the polypeptide chain, if you ever heard of an organelle called the rough endoplasmic reticulum or rough ER, that folds the proteins into their secondary and tertiary shapes, and then it has to go to the Golgi apparatus, which does some of this finishing work. And then after it leaves the Golgi apparatus, then we can call it a protein. Now it's mature. So don't focus on that yet. I'm just trying to mention some things that you've learned in different classes. So hemoglobin in our red blood cells is a great example of this quaternary structure. Okay, it's different chains of proteins that have been made, and then they combine in a specific way to make this larger unit that our body needs. Okay, and sometimes it's nice just to see everything on one page instead of seeing individual slides. So again, primary protein comes from the DNA, and it's okay, this is the order that you put your amino acids in. It's almost like if you had instruction manual on how to make a beaded necklace in summer camp or in some art class or something like that. It would just tell you which order they go in. That's the primary structure. But our bodies are always going to fold them. And when nearby amino acids attract each other, we see characteristic spirals that we call these helix, and these kind of ripples that we call this pleated sheet. And then, if this amino acid were to attract to this one, it would kind of crumple it, because they're not nearby, they're far away. And then we kind of get this crumpled globular shape that's called the tertiary structure. And all proteins exist as tertiary proteins, and some of them will combine to make something larger, and that would be a fourth level protein. Okay, we'll learn this year that what can happen if you were to, say, heat up a protein. What would happen is you would take these more complicated layers and it would unravel and it would look like this again. Okay, high temperatures, and that's called denaturing, going from right to left on this picture. It means you lose the shape. Think about if you have a key to get in your house. If the shape gets messed up, it doesn't work anymore. So this shape is very important as we go up through these levels. But if we undo this shape, which can happen with high temperatures, it's what happens when you cook an egg. It's why the gooey part of the egg becomes an egg white, is you're actually unraveling the albumin protein, and it's becoming this primary structure again, and they don't work. So we'll learn more about that. So just focus on the differences between primary, secondary, tertiary, and quaternary protein structures.